The name of Allah, the most beneficent, the most merciful. My name is Dr. Yasir Waqas, and today I am going to present exosomes as novel regulators of reproductive physiology. So, when we talk about the cell to cell communication, we have different transport mechanism in which the membrane transport through simple facilitated active transport and passive transport is on the top of the list. The second way in which the cells are communicate are through the junction complexes, in which we have three kind of junctions, like simple junctions, complex junctions, and desmosomes. The uh, the way of communication through extracellular vesicles is an innovative one, in which from 2016 to 24 there are a lot of research in the field of extracellular vesicles. So they have a direct role in the cell to cell communication. When we talk about the biogenesis of these vesicles, actually they are enveloped with the lipid bilayer and they are reserved in all mammalian cells. The research studies shows the involvement of EVs in different mechanisms like long-term sperm storage, carcinogenesis, cardiovascular diseases, and metabolic syndrome. When we talk about the size of these particles, there are certain three categories: the exosomes, multivesicular bodies, and apoptotic body. The exosomes are 30 nanometer in size, the microvesicles are 100 round about 100 nanometer in size, and the apoptotic body are around about 1000 in size. When we see the biogenesis of these vesicles, actually the exosomes, which are 30 to 150 nanometer in size, they are actually enter the cells through the phase 1 pathway, which is endocytosis of these particles. After processing with the Golgi apparatus and nucleus together with the rough endoplasmic reticulum, it appeared as early endosome, then formed into multivesicular bodies. The multivesicular body is that kind in which you have more than six exosomes in that single unit of the MVB. After detaching with the after in contact with the membrane, the exosomes are shedded outside the cell, and they are then further communicate with the other cells through that way. On the other hand, microvesicles are bigger in size. They are involved, are they are released through the direct shedding from the membrane. When we talk about the apoptotic body, and you can see in the third and the right end of that angle, there are apical bulb. Sometimes the whole cell goes out, and sometimes half of the cell are released in extracellular space. So the area of the research is the when we talk about the identification of extracellular vesicles, we have different like prostosomes, immunosomes, exosomes, and oviductosomes, which are already the prostosomes are identified in 2016. The same like immunosomes and exosomes, and there are certain type of organs which are involved in the release or shedding of these particles. In the 2016, I have first paper on the identification of the exosomes in the epididymis and vaginal section of the hen. So the main focus of the research is long-term sperm storage. Like in previous slides, I have discussed that the EVs has a direct role in the long-term sperm storage. So our main point of the discussion is that sperm storage is obvious in many animal species. You can see the sperms are stored in the hen for about 15 days, for 24 hour to 48 hours in the human, and 1 to 5 years in the case of turtle. So what are the microenvironment which facilitate the long term sperm storage in different animal models? Either there are the factors coming from the sperms are the main reproductive tract, are the other are there are certain criteria, are the factors which are in sportive in the female reproductive tract. So the first research about the cellular evidence of exosomes in reproductive tract of Chinese softshell turtle. You know that oviduct is a location of egg production, fertilization, and sperm storage. While its secretions has broadly attributed towards different physiological functions. We examined the ultrastructure of oviduct epithelium and glands in relation to secretion, particularly with the exosomes in nature. When we talk about the segment of the oviduct starting from infundibulum, then comes magnum, isthmus, uterus, and vagina. So the immunostaining of the isthmus, uterus, and vagina during January shows the positive expression of CD63 antibodies, because these antibodies is against to identify the presence of exosomes in the epithelium and glands. Immunostaining was again positive during the May, but the expression was little bit moderate as compared to January. The same case in the immunostaining of the isthmus uterus during September shows the positive expression in all segment on the epithelium as well as on the glands. While during the hibernation period in December, it also again shows a positive expression. So, nutshell is that you have seen a positive expression starting from the January up to the December, but the moderate level of expression was during the hibernation period, in which the reproductive tract initiate the next cycle of the fertilization. While the images also the data was also confirmed with the transmission electron microscopy, you can see there are a lot of secretions and exosomes around the sperms in the epithelium and glands during January. The same results were identified in the all segments of the reproductive tract, and the exosomes and secretions were there on the epithelium as well as on the glands of the segment of the reproductive tract during December. So during December, you have exosomes near to the sperms. You can see in the second micrographs, you can easily see identify exosomes near to the sperm and together with the microvesicles and secretory vesicles. So when that was all about the data coming from the turtle. Now if you see in the uterovaginal junction of the hen, because it is involved in the storage of sperm for 15 days, you can also see the micro vesicles and the exosomes in the epithelium and of the cilia, because the epithelium contains two type of the cells, ciliated and secretory cells, while you can easily see in the exosomes are present on the epithelium of the segments of the uterovaginal junctions. Uh, the the secretory vesicles are also you can identify near the ciliated cells, near the secretory cells, together with the circulation. So this is a micrograph indicating the multivesicular body. In the introductory slides, I have told you the early endosome is converted into multivesicular body, which contains lot of exosomes, and these exosomes come out when it got contact with the epithelium of that. So concluding remarks are that we have found the ciliated and gland cells are involved in release of exosomes. This both shows positive expression for CD63 as a membrane marker in different months, and these data is also in line with the evidences provided by the transmission electron microscopy. So there are different if you analyze the protein nature of these micro vesicles or the exosomes, there are a lot of type of proteins, and majority contains the antioxidants in that one together with the protein. So that's all about my publication related to the current topic. This is my research group that is working on the microvesicles and exosomes. and we are hopeful that in the future we are directly involved in the sequencing of the all the genes which are controlling or which are involved in the release of these particles. Current study in 2024, which is published in the Journal of Physiology and Biochemistry with that title. Thank you very much. Any question?